So hello everyone, I am Shrabasti and I am an ISERC alumnus from the 2014 batch, and we graduated in 2019, so I think that's why a lot of you might not know me by face. And currently I am a PhD student at the Institut de Neurosciences de Timone in Marseille, France. And I am a part of the COMCO team, which stands for Cognitive Motor Control team at this institute. And basically, as the title of the talk suggests, I work in work studying brain, and specifically I study the cognitive functions in brain. And more specifically, I study the cognitive motor control in brain. And I'm interested in cognitive functions because these are highly complicated computations that happen in our brain, and which basically start from as simple as electrical signals. And that is really, really fascinating to me. So before I go ahead, the primary question that always struck me was, this is not a question to me. I know that humans are different, definitely. But why? Why are the humans different? And if you, apparently the humans should not be different, right? Like the physicalities of humans are very like average. But the answer to that is the brain. So this is very well shown in this very old ad from our childhood when we were in standard 9th or 10th, and this is a Mentos ad. And I'm not sponsored by Mentos, by the way. So don't like think that I'm promoting Mentos or anything. So this is the Mentos ad, and it's a fun ad. It's a very creative ad, I think. Tell me if the audio is.
Yes, audio is visible.
Is there? Perfect. Yeah, perfect.
Good. Okay, so do you all remember this ad?
Yes.
Yes, Didi. Yeah.
Yes. So I feel that this ad is like, obviously it's not scientifically accurate, as in obviously there was no Mentos in prehistoric ages. That is not what I wanted to prove here. But the point that I wanted to show you is that even in popular culture, it is very well established that humans are different. So see, here also they showed that the apes, the early humans, they were controlled by donkeys, which is also not true. But they could be, right? Like they are very average. Humans are very average. Why couldn't they be controlled by a donkey, first, so to speak? But they are not. And why? Because there is something which happens unique in our brains, not because of Mentos. I'm not saying that. So what does it mean by dimag ki batti jala de? So there is something different going on in our brain, obviously. But what? So the things that are different in our brain are the very complicated thoughts, emotions, reasoning, jotting down information to solve different problems, and all of that. Paying attention, forming memories, using those memories to make our future experiences better, all of that. So all of these collectively are known as cognitive functions. So cognitive function is anything which any action that you perform due to the complex computation of a lot of different information that you receive, that your brain receives, to make your life more comfortable, more meaningful, more easy, okay? And what is cognitive neuroscience then? That is just studying the neural processes, the pathways which produce these capabilities. So before going to cognitive functions, I want to go through briefly by the basics of neuroscience. So I start by the structure of the brain. So one reason why the humans are different than the other living creatures is the structure of the brain. You will see why I say that. So if you see a human brain at its embryonic form, when the embryo is 5 weeks old, you'll see such a structure, which does not look like the brain that we are very familiar with. So this is called a reptilian brain. This is the very core structure which is needed, which is basically responsible for our survival. So these structures are responsible for all functions like eating, sleeping, maintaining the circadian rhythm, and mating, all of that. And this is the spinal cord. This is the hindbrain, which later develops into the cerebellum, the pons, the medulla oblongata. This is the midbrain. This is the forebrain. And the forebrain has two parts, the diencephalon and the telencephalon. And you'll be surprised to know that this telencephalon, this tiny structure, this is basically in later part in our adult human states, this is the 80% of the brain, and this is not required for survival. When the embryo grows to 13 weeks, you can already see that the telencephalon has grown quite a bit, and it has already covered the diencephalon. If we go further to 26 weeks old embryo, it has grown even further, and it has now covered even the midbrain. And you can see these fissures and folds that have started appearing in the cerebral hemisphere or the telencephalon. And this is for increasing the cerebral hemisphere surface area, right? And now this is the dramatic form of the human brain at the birth of a human child. So now you can compare this structure to this big, huge, humongous structure, which is so complicated and so densely packed. And all of this is just for the fine tuning of your life. It's not related to the survival instincts that you have. So basically, having better vision, having better hearing skills, having better social skills, having better communication skills, all of that happens here. Okay, and that is one of the reasons why humans are different than the other species. Okay, so we know that in our nervous system, there are two parts. The first part is the central nervous system, and the other part is the peripheral nervous system. And the central nervous system basically comprises of this thing, this huge thing, and this thing. So the brain and the spinal cord. And the peripheral nervous system are the nerve extensions that go to your peripheral organs, like the limbs or your body, the trunk of your body, for mediating the information that comes from this or the spinal cord. So how is the information transmitted from the environment to the brain, and again from the brain to your executing organs of your body? So it is communicated by electrical signals. Now we all know that for electricity to flow, the basic things that is needed is a positive and a negative terminal, right? And basically these are positive charges and negative charges, aggregation of the positive and negative charges, and a gradient. And how is the gradient formed? By an insulator. So in case of neurobiology, the insulator are the cell membranes or the tissue, the tissue of the neurons, right? And now to conduct the charged particles, we also need some channels here, and that is exactly how electrical signals are communicated from the outside of the neurons to the inside of the neurons. We'll see how. First, I want to say you that the resting state of a neuron is characterized by its resting membrane potential, that is a constant potential of minus 70 millivolts, which is maintained. And how is it maintained? So the first thing is that the cell membrane has a large number of negatively charged proteins, which are formed inside the cell and trapped within. Also, there are these structures called as sodium-potassium pump, and these use ATP to transfer 3 sodium ions outside for every 2 potassium ions. So you can see that more positive ions are transported outside than inside. So definitely the outside of the cell will be more positive than the inside. So this thing accounts for approximately minus 50 millivolts of the potential difference. Okay. And apart from that, there are these leak channels, potassium leak channels. So since there is a higher concentration of potassium inside the cell, these leak channels allow some potassium ions to go outside. And also, when there is higher concentration of positive charge outside the cell, there are some potassium channels which transport the K plus ions inside. And this basically, these two cycles, the drive of the to balance the chemical gradient and the drive to balance the electrical gradient, meet at a point which is called the electrochemical equilibrium, right? So the point where there is this electrochemical equilibrium, that is the resting membrane potential, and that is around minus 70 millivolts. Okay, now we know how the resting membrane potential is maintained in a neuron. But what happens when a stimulus comes? How is the information transmitted? So basically, when there is a stimulus, say this is an excitatory stimulus. Excitatory means that the membrane potential will go positive. Okay. Suppose the stimulus is very weak. What happens is there are positive ion inflow in the across the membrane. For example, let's say some calcium 2 plus ions get in the membrane, and the membrane potential rises. When the membrane potential rises by a little but does not reach a particular value, which is called as the threshold, the stimulus fails to initiate an action potential or to transmit the information. And again, the resting membrane potential is restored. But if the stimulus is strong enough, this threshold is reached. And at this threshold, there are these guys. These are voltage gated ion channels. Basically, this is the sodium voltage gated sodium ion channel, and this is the voltage gated potassium ion channel. And these are initiated at this threshold. So what happens? These gates open, and there is an inflow of sodium plus ions. Why? Because there was higher concentration of positive ions outside, right? And also this this ion channel also open, but it does not open completely, or the opening is very slow. That is why a very small amount of K plus ions, which is higher in concentration inside the cell, can go out. So after so not only one sodium ion channel is present, right? A lot of them are present. So there is a very fast phase of depolarization, that is the rise of membrane potential because of the Na ions coming in. And after a certain period of time, even the potassium ion channels open completely, and the potassium ion can go out now. And when the membrane potential reaches around plus 40 millivolts, this ion channel gets deactivated again, and this thing gets completely open then, and there is again a period where the membrane potential falls, and this is called as a phase of repolarization. This fall even goes beyond the resting membrane potential, and this period is again known as hyperpolarization. And then this hyperpolarized neuron is brought back by these structures again to the resting membrane potential state. So this is basically how an action potential is formed. The basic idea here is in electrophysiology, in extracellular electrophysiology, or in cognitive sciences, the dynamics of this action potential is not something that we care a lot about. The thing that we really care about is at which period, at what time was this action potential initiated, and at what frequency. So suppose an action potential was initiated at 2 milliseconds, and again at 6 milliseconds, and again at 8 milliseconds. This is what we want to keep a track of, not the dynamics of what happens here. So basically, we just approximate the entire thing by just one binary digit, that is 1. So this is a state 1. And every, say, 10 milliseconds time period, if there is no action potential, that is a 0 state. So this is a neuron. We all are very familiar with the structure of a neuron. This is the cell body. These are the dendrites. This is the axon, and this is the axon terminal. And so the dendrites get the inputs from other neurons. There is an action potential generated throughout this axon, in the cell body and throughout the neuron. And then the action potential basically triggers some synaptic vesicles here to fuse with the cell membrane here, and the neurotransmitters or the electrical signals are transmitted to other different neurons. So there are multiple different inputs, and there could be multiple different outputs also. And all of this is very similar to the computing that the computing approach that we use in our computers. So basically, every time there is an action potential, we also call it as a neuron firing or a neuron spiking. We just say that it's a state 1, and every fixed intervals of time when there is no spikes, we say that these are state 0s. And this entire thing, this entire dynamics of spiking throughout a certain period of time, this is called as a spike train. Okay? And this is the unique code that one neuron gives to another neuron, and the other one to another, and so on. And these unique codes are basically then processed, and similarly as is done in computers. And then these signals are then made sense of. So I'm really happy to be here today because I feel that I have really come a full circle. So on 31st January 2016, my journey with cognitive neuroscience basically started. It was the second ISERC IO. We had just started ISERC IO, and in the second ISERC IO, I basically presented what brain GPS is, and I was so excited by the concepts of cognitive neuroscience. And I feel really emotional today that I'm again speaking in ISERC IO. So basically, I came across the Nobel Prize winners of 2014, who were John O'Keefe, May Britt Moser, and Edward Moser, and they got the Nobel Prize in medicine and physiology because of the discovery of brain GPS. So it sounded very interesting to me. Brain GPS, we are very familiar with what GPS is, but brain GPS was very like a catchy thing for me. And then I read up a little about it, and the approach really caught me. I was really fascinated by the approach that is taken by cognitive neuroscience. So I will just explain what brain GPS is to you also. So what is brain GPS? As the name suggests, there are certain structures in the brain which encodes the spatial location in which we are currently. For example, if I'm sitting in this room, there are certain neurons in my brain which encodes for this particular spatial location. Okay. It also helps in the formation of episodic memory. So to explain what episodic memory is, I again want to show you a video. And this is from a Pixar movie, and I'm a big fan of Pixar movies. They have tremendous animations. And so you can also watch it along with me. I hope there's no ad here.
Grammarly can help you write quickly and confidently.
Yeah, I'm sorry for the ad. Okay, so this is from the movie Ratatouille, and you will see what is meant by an episodic memory. Let's watch the video and I will explain what it is.
Ratatouille? That must be joking.
Okay, so did you understand what episodic memory is? Can someone explain from maybe this video?
As if the
Like the
flashback
same
triggered by some incident.
Yes, exactly. That is what it is. So basically, he had the ratatouille, and he found the taste to be very familiar, and that triggered some memory. So basically, there is a complete encoding of space, context, time, what events took place at that period of time in that space, and all of that is an episodic memory. Okay, and so for forming this episodic memory, we definitely need the encoding of spatial location. We also need the other sensory information, for example, association of taste, association of smell, association of some auditory cue, etc. And we also need a sense of time, right? So there should be some mechanism through which our brain tracks the time. So brain GPS is basically the collection of all of these functions, and which structures in the brain are responsible for the completion of brain GPS are these: the entorhinal cortex, the hippocampus, the subiculum, the dentate gyrus. All of this is called as the hippocampal formation. So does anybody
Hello.
have any question?
Yes, Didi. Actually, Tapovan wrote a question
Yeah.
in the chat box. So he's asking a catalyst for a lost experience, maybe.
A catalyst for a lost experience. Yeah, that is an interesting analogy. Actually. Yeah. So it could be so it is not necessarily supposed to be a lost experience, but yes, definitely a catalyst for remembering some experience, right? So it is not necessarily supposed to be something that you have forgotten and it's long back, but some things, for example, you are listening to a song and you remember certain experiences, you remember a certain trip, and it's not necessary that you have forgotten about that trip, but it's just a trigger for remembering that trip, right? So that is what episodic memory is. Okay. So coming back to the brain structures which are responsible for the encoding of space-time and all of that, it is the hippocampal formation, which comprises of the entorhinal cortex, the hippocampus, subiculum, and dentate gyrus. And so this is a disclaimer. Here I'm talking about a rat brain, okay? Because most of the hippocampal formation studies are primarily done on rats or mice. Or mice. So this is a three-dimensional image of a rat brain, and this pink thing, this is the hippocampus, and if you take a cross section of this, it looks something like this. And so this thing, this banana shaped thing, this is the hippocampus. It has three areas, CA1, CA2, and CA3. Here is the entorhinal cortex, this area. Okay, this is the this bright colored thing. This is the dentate gyrus, and this is the subiculum. And all of these in some form communicate the information of space and the associated sensory information. So basically, the context is encoded mainly in the entorhinal cortex. For example, in lateral entorhinal cortex, object-related inputs are sent to the hippocampus. And hippocampus is the major site of integration of all the spatial cues. So it encodes for space and also integrates all the information from the cortex to form the episodic memory.
There is a question
Yeah.
by Olokendu De that he is asking that is episodic memory has any particular time interval?
No, it could be as small as say some few seconds to minutes. It is really as long as hours. It cannot there cannot be a very accurate memory for an hour-long event. But yeah, seconds to minutes.
Okay, so Akash has a follow-up question. So he's asking, is episodic memory can be removed / forgotten by any way? So if possible, what is the mechanism?
Okay, so for this, basically you have to understand how memories are formed. So there's a process called synaptic pruning. I don't want to go a lot into the details, but I'm crudely explaining which is related to the question. So what happens is when we are of young age, there are a lot of neuronal connections which are there in our brain. But as we grow older and as we use some connections more than the others get pruned. So the other connections get removed. And the ones that we use more get stronger. And this process of one connection getting stronger because of repeated use is called plasticity. And like, this is a very crude explanation, okay? This is not a very, scientific or very accurate explanation, but this is as crude as, which is required for understanding the concept. So plasticity is basically the repeated use of certain neuronal connections, and that is how memories are formed. So I don't know if you have heard about this. While growing up, my father used to always say that brain is like a tape recorder. More you play certain songs, those certain songs get more you hear the songs better. So similarly, if you have had some experience, and say if it's a very good experience which you want to remember, you repeatedly remember it and cherish it, so those memories get stronger. And say you have had water 2 minutes back, you don't want to remember that, right? That's not a very crucial piece of information which you want to store in your brain, so you forget that. And that is how episodic memories could also be forgotten. But the memories which you relive or remember repeated the number of times, those memories get more strong. Yeah. Did that answer the question?
Akash?
Yes, Di. Okay, Di. I got some explanation.
Okay. I will come to the memory consolidation part just in a bit. Okay. So yeah, so this is the hippocampal formation, which is important for the encoding of spatial location and for formation of episodic memory. Now, what exactly are the place cells and grid cells? So this is the Nobel Prize press release from 2014. You can see this is like half of the Nobel Prize was given to John O'Keefe and the other half to Maybrit and Edward Moser. And so this man, he discovered place cells, which are in hippocampus. And these two people, they discovered grid cells, which is in entorhinal cortex. So what is a place cell? Place cells are any cells in hippocampus which are mainly in CA1 or CA3, less in CA2, which fire, you remember what firing is, right? Generation of action potentials. So cells that fire when you go to a particular location. So this is a big environment. This is the rat. It travels through all of this part which are gray, but whenever it comes to this location, this cell fires, and this is a place cell. Okay. Grid cells are also similar cells, but these have repeated receptive fields. What does that mean? So for example, this is a grid cell, this blue colored thing. And whenever the rat goes to any of these blue areas, the cell fires. Okay. And the thing that you should notice here is that this pattern is very regular. It almost forms a regular hexagon, right? And the hypothesis is that the grid cells in some form give the idea of how much distance was traveled. So if a grid cell has a regular pattern, and if it fires when you are here and when you are here, or when you are here and when you are here, so basically somehow you know how much distance you have traveled. You can encode the distance, and that gives you a sense of along with the place cells, that gives you a sense of direction and distance. So that is a very crude explanation of how space is encoded in our brain. And so yeah, this thing is all these studies are primarily done in rodents, which is mice or rats, but later studies have also found that similar structures, similar to the hippocampus and the entorhinal cortex, and similar cells like the place cells and grid cells also exist in human brain. Okay. Okay. So as I said, I come back to the evidence of memory consolidation in hippocampus. So These are spike trains. I explained what spike trains are. So at all of these times, so for example, here we have 13 different cells, okay? Every single color represents one neuron. And so the neuron 1 fires at this place, at this place, at this place, at this place, at this place. And this is the spatial map of the rat traveling in this environment, okay? And you can see that this cell fires first, and then this, and then this, and this is arranged in an ascending order in time. So the cell that fires first, and then the one that fires after, and so on. Okay, and there's a sequence, right? Going from blue to red. The interesting thing that was observed was that after the rat traveled in this area, and after that, when the rat had brief durations of pause, in this window of time, there was a compressed replay of the reverse replay of the events. So as you can see, there was an ascending order of sequence from cell 1 to 13 of the firing of the cell 1 to 13, and now you can see a descending order of firing from cell 1 to 13, right? So the first cell fires, the last cell fires first, and the first one at the last, and so on. And so the idea, again here, the hypothesis is that this reverse replay just after the task helps in the consolidation of the memory. So since the entire sequence is repeated even when the rat is not traveling in this space, it shows that there is some way in which the brain is encoding the experience that it just had. Also, there are some studies which show forward replay. That is, suppose if you have been used to traveling in this piece of environment for, like, say 10 days, later in the days, you will find that just before the rat starts the task, there is a forward replay. So the first cell to the 13th cell fire in an ascending order, just like it would in the actual space, but in a compressed time zone. So you can see here it is in milliseconds, 0 to 25, and here it is in seconds. Okay. So this is some evidence of memory consolidation. So basically, once this pathway is reiterated, this memory is supposed to get stronger. Okay. So for studies as such, as I said, rodents were used, rodents are generally used. But in our lab, in the lab that I worked for my summer project, in my second year and in my fourth year, the subjects that were used were male long Evans rats. Long Evans rats are lab rats. They are specifically bred for the experiments in labs, and they have very high cognitive skills. They're very smart, and they are housed in. So this is basically to maintain their natural biological or circadian rhythm, 12 hours light and dark cycle. And obviously we don't want to torment the rats which are younger. So the rats have to be of a specific age before they are used for the experiments. And then they also need to get used to the experimenter. So that is what we call as handling. So basically we get used to the rats, and the rats get used to us for around 2 to 3 weeks. And then the rats are implanted with electrophysiological hyperdrives. I will say what hyperdrives are in a bit. So these are hyperdrives. Basically, here we have these are custom built. These can differ in configuration, but in this particular image, here we have 16 tetrode hyperdrive. Tetrodes are, as the name suggests, tetrodes is a combination of 4 different electrodes turned together, and every electrode is passed through these canals, and these canals are then screwed in through this structure to the brain. So during the implant surgery, what is done is a part of the cortex is removed. A part of the skull is removed, which corresponds to the position which is above the hippocampus. And we know that by previous experiments and by coordinates, and there are some like sutures and sulci on the skull, by which we can locate at which point we want to introduce the implant. Once that is done, the rats are woken up, the surgery is closed and the rats are woken up. And after the rats recover from the surgery, these hyperdrives are driven slowly inside the cortex. And as you could see here, the hippocampus is quite deep. So suppose if you want to reach this part, you have to drive the tetrodes quite a bit. So that takes a little bit of time. And once you reach the hippocampal layer, you know that by the signature electrophysiological signals. So I said what spikes are, right? But there is also a thing called local field potential. That is nothing but that is the summation of all the extracellular activity, low intensity extracellular activity that occurs around a neuron. Okay, so every layer in the cortex or in the brain has a signature electrophysiological signal. For example, in the hippocampus, there are very like high frequency activities which are called as sharp wave ripples, and also there is this very uniform frequency, 6 to 10 hertz frequency oscillations that is very like visible. Like I don't know how I could get back to it after I finish presenting if anybody wants to know in greater depths. Okay, so for now, just let's keep in mind that these are some very regular frequency oscillations that happen in our brain because of the structure and the orientation of the neurons, and these are very specific to certain layers. Okay, and when we drive these electrodes or the tetrodes in, and when we see those signature electrophysiological signatures, we know that we have reached one particular area. And so once that is done, once we reach our desired area, we get two things: single unit spikes, which I have already explained, which are nothing but the action potentials, and these local field potentials, which are the summation of different neuron activities. And these are the low intensity signals. So basically, when you have the raw signal, you filter that in two parts. You get the low frequency signals as local field potentials, and the high frequency signals as the single unit spikes. And once you have these single unit spikes, what you do is you identify which spikes belong to which neuron, and for that you have I can show that to you later once we are done with the presentation. But basically what you have is you have a screen in which there are different clusters appearing, and when there are two different neurons, you will clearly see two different clusters appearing very distinct from each other. And from these neurons, when you identify these neurons, you get the spike trains. And then from those spike trains, you can just see where or at which moments are the neurons firing, and you get such a place map. So this is a linearized position. So this is basically from 0 to 1 in a linear maze, for example. And when the rat is traveling in all of these areas, the neuron does not fire. And in these highly colored areas, the neuron fires. And this is a place cell. Okay. And local field potentials, from that you can identify which area it is, as I said. And also you can find some interesting events even in those local field potentials. For example, you can detect the sharp wave ripples. You can also get some information from the nature of the sharp wave ripples and the specific modulating oscillatory signals. So that is what is done in electrophysiology and in this approach of cognitive neuroscience, basically. So this is what I was doing in my second year summer, also in my fourth year summer, and also in my dissertation. Now, I really wanted to get a little bit more closer to humans, the human cognition. So for that, I applied to a lab which works with monkeys. And as I said, we study the motor cognition. So basically, when you, if I hold this phone and if I want to pick this phone up, I very accurately pick this phone up. I don't overshoot the position, or I don't undershoot the position. I very thoughtlessly and effortlessly, I can pick this phone up. But this is a very complex activity that actually happens due to the coordination of multiple different areas in our brain. And that is what we basically study. So for my current study...
before that, hello?
Yeah.
There are some questions actually. So first, Olokendu, he is requesting that please repeat about single unit spike a little bit.
Okay. So this is single unit spike. As I said, the transfer of electrical signals in and out of the cell membrane or the neuron creates this very rapid increase in membrane potential. And this increase in membrane potential, this has a very typical shape, and this is called as the action potential. And we approximate the action potentials as spikes. We do not care about the shape that much. I mean, in some studies we do, because this shape actually tells us about the nature of the cell and which kind of cell it is and all that. But for simplicity, for now, let's just ignore the shape of the action potential. Rather, just we will concentrate at which point this peak occurs, right? So we just approximate it by these lines, okay? And these are spikes. So whenever there is an action potential in one neuron, that is a spiking neuron. Is it clear?
Okay, Diti.
So there I have a question also that in single unit spike, that you shown this thing at like as a bit like thing, that there are 8 unit you are taking, like as we take in our computer. So this is just an assumptions for dealing with like bioinformatics things, or it is just really happen in our brain also? This is just like
so it is very difficult to say the exact mechanism by which our brain functions, right? We can just like go by approximations and assumptions. We are still not at that state of art technology that we can say what exactly happens in our brain. But yes, this could be one of the ways in which our brain processes the information. It could be a 8-bit thing, it could be a 16-bit thing, it could be a 32-bit thing, and so on.
So essentially, like, is this a way of just representing the information?
Yes. This is something which is used in artificial intelligence and for making prosthetic organs and things like that.
Okay. And Didi, there are two more questions in the chat box. Shall I take? Will you take them now?
Yeah, sure.
Okay. So the next one is I'm going from bottom to the top. So the next one is from Akash again. So he's asking, as there is a potential difference, there will be some changing electric field, also changing magnetic field. Is does the neuron show any response? If yes, how do neurons respond to the same?
Okay, so the first thing is that you have to understand that the electrical potential gradient, that is not a very strong one, right? So it is in millivolts. So even if there is a magnetic feature associated with it, but it is not supposed to basically disrupt the electrical functioning. Okay, that's the first part. And the second thing is that exactly this thing that since there is an electrical charge dynamics, there are some magnetic properties which are related and which are basically exploited for a lot of imaging techniques. So you might have heard about functional magnetic resonance imaging or magnetic resonance imaging, right? So essentially, the different magnetic properties of different substances create a very vivid image for the functional magnetic resonance imaging and MRI. But yes, for the physiological functions, the physiological functions do not get disrupted or do not get affected very significantly because of the magnetic field, because it is a very low intensity field.
So the next question is from Aritra. He's asking, at the next evolution of human, what will be the adaptations in our brain?
Well, that is a very, speculative thing to say, right? I think like we are already evolving very rapidly now. As you might have heard, the generation gap now has reduced to like 2 years. So with every coming generation, the brain gets more equipped, so to speak, for in terms of cognitive functions. And evolution, we have to remember that evolution is a continuous process, and to see a very like drastic and very dramatic change, I think that's going to take a while.
Okay, and for now we have this one last question from Priyadarshi. So he's asking, do place cells collect information and make them to a grid by sending information to grid cells?
Yes, there is a forward feedback loop that goes on between the hippocampus and entorhinal cortex. So information from the, let's see, okay. So information from the entorhinal cortex comes to the hippocampus. For example, let's say from the LEC, from the lateral entorhinal cortex, some object-related inputs come to the hippocampus, and then after this, the hippocampus might send some information to the MEC, that is the medial entorhinal cortex, for the better encoding of the grid cells and so on. So there, it is a two-way thing. Yeah.
Okay, no more questions, I guess.
So let's move forward. Okay. So we are changing tracks a little bit. For my current study, the approach, the animal model, the questions, all of these are very different from what we were looking at till now. So we use monkey models, and we want to study the visuomotor cognition pathway. So basically, when we are seeing an object, processing the visual information, and then sending some execution outputs to the motor cortex, and then in turn moving our hands or doing some things in response to the stimulus. That is what we want to, that is the aim of the study, that we want to study that network, so the visuomotor parietal cortex network. Okay, so for that we have implanted these very high density Utah arrays. These are called Utah arrays. They look like this, and these look like matrices which are made of either 10 by 10 electrodes, that is 100 electrodes here. In every row and in every column there are 10 electrodes, or 6 by 6 Utah arrays. These are the smaller ones. So every row and every column has 6 electrodes. And these are implanted in certain cortical areas. And the cortical areas are marked by fMRI. So first, the monkeys were subjected to fMRI, and the brain regions were marked. And using those information, the surgery was performed, and these arrays were implanted. So here you can see this array. This is the big array, the motor array. And this entire thing, this entire head stage, this is for all of these 4 arrays. Okay, so it has 4 different arrays, 2 in the visual cortex and 2 in the parietal cortex. Parietal cortex transmits the information from the visual cortex to the motor cortex, and it also gives some additional context to the motor cortex. This is basically very crucial part for motor control. So motor cortex transmits the information about which part of the body has to be moved, and this is the area which is responsible for making the decisions, or controlling the motor cortex, so to speak. Okay, so the task is such. We call this as a landing task. So basically the monkey sees targets at certain positions, and it has to move its hands to reach those positions. So here you can see this is the camera through which the eye is recorded, the eye gaze is recorded. This is the screen on which the target appears. So this white spot, no, sorry, the pink spot, that is the target, and the white spot is its hand feedback. So when the monkey knows that when the white spot is here, the hand is here, and it knows that it has to reach this white spot to this pink spot to have a successful reach. Okay, and why? Okay, so I have never talked about why the animals do the tasks that we want them to do, right? So basically, the animals are trained with some food deprivation or with some water deprivation, and when the animal does some required, the response is what we want, the animals are rewarded by either water or food, depending on what they were deprived of. So in our study, we deprived the monkeys of water in the weekdays. I mean, it's all according to ethical standards, as in we are not allowed to, it's rather better to put it in this way, that we are not allowed to manhandle or mishandle our animals. We have to take very good care of our animals. But and that's why we are supposed to make sure that the animal gets a minimum amount of water at the end of the day in some way or other. But as long as it is above a threshold, the water requirement is above a threshold, we deprive the animal of extra water, and the animal works for getting water rewards. Okay, and this is, so basically the hand is supposed to reach the target. That is it. And the task is as such. So basically there's this hexagon which appears on this flat screen, and every time a target lights up, as is in the pink case, the hand is supposed to reach the target. And in my task, what the monkey is supposed to do is that it has to form a sequence of trajectories. So for example, this target lights up, the monkey has to reach to this target. Then after this target is reached, this target lights up, it has to reach here, and then this, and then this. And then he gets a water reward. Again, this is the first trial. And again, this one would light up, then this, and then this, and then this. And then again, a water reward. That is the target. Okay, so the target appears, he sees the target, reaches there, and another target lights up, and so on. That is how it proceeds. The thing that we observed after doing this task was that even though the distance that the hand travels is similar for all of these reaches, there are certain reaches which have more spatial variability in terms of the trajectories that the hand makes, right? Here you can see it's more spread out than these ones, or than these ones, or these ones. So we want to quantify this, and we want to see what actually, at which level is the neuronal activity getting different, which leads to such variability. There is no reason for variability in very similar, similar paths, but still we see these variabilities. And this is the most simplistic case that I'm showing. We have also seen that when the monkey is making paths like this and this one, say, these are in the same direction, but even then this path is very different to this path globally. So we want to see why these differences occur and at which neuronal level do the differences come from. And is it a neuronal process that leads to these differences, or is it a kinematic process that leads to this difference? So by kinematic process, I mean that if suppose one particular direction is more difficult for the monkey to make, the trajectories could be more different than the others, okay? So the possible origins of such variabilities could be a complex target, as we said. This target being more difficult could be a reason. Also, anticipation of the sequence. So you have to remember that the monkeys are trained. So it's not only, it's not a task done in one day. So the same task is repeated and repeated again every day throughout like months. So after a point of time, the monkey already knows the sequence. So for example, I said that the monkey is supposed to make such a sequence, right? 1, 2, 3, and 4. And when it has made this sequence, it does not know where it was supposed to go. So it could go to any of the 6 directions. But when it has made this these 2 paths already, it has already been trained with the task. Now it can anticipate this task, this trajectory, and this could also lead to a less variability. And also the very physiological and the problem with any animal experiment, that is the level of motivation varies from day to day. So one day there could be some problem in the animal facility because of which the monkey could be less motivated, or suppose it's more hot someday than some other day. So that could affect the level of motivation, and it could pay less attention to the task, and that could lead to the variability. So it could be physical variability sources. Also, it could be neuronal variability source, as in there could be something, some process in some of these areas, the motor, the parietal, or the visual could go wrong, because of which the variabilities could sum up, right? So we basically want to see if it's a top-down process which leads to such variable paths, or if it is a bottom-up process. Bottom-up as in starting from the kinematic variables to the neuronal variables, or the other ways. So that is the current study that I'm doing. And after I do that, I want to relate the variability that we would observe in the neuronal level across the areas to see if one area is, if the variability in one area is leading to the variability in another. So if it is occurring before in time than the other. And so we could essentially track down the pathway that is leading to any variable movements. And the prospect of this work is that if we can analyze and track down what leads to variable movements, we could maybe in future make more, like we could improve the motor skills of certain patients, or we could basically use some behavioral exercises to increase the motor control cognition. So, and after that, we are also planning to do some microstimulation studies, and I have not talked about microstimulation yet. So basically what we do is we have already these Utah arrays implanted in these areas, right? So we pass very low currents to simulate the information that would otherwise come from any other part of the brain or from the sensory organs, okay? So the brain area would receive the stimulus as if it is receiving from its natural pathway. That is microstimulation. And we want to do some microstimulation experiments in these areas to see if, suppose if we find that this, the variability in this area is occurring before this, and if, I mean, before in time, so we could do microstimulation experiments in this area to see if if similar variability is getting formed in these areas as well. So that is the idea. And we are yet at a very, like, we have collected some data. We have to analyze the data yet. So yeah, so that is what I'm doing right now. And to end today's session, I just wanted to give a flavor to you to show how studying these cognitive functions could actually practically help people. So this is a recent paper. And here also, they have used microstimulation techniques. So basically, they have made a prosthetic limb. So this is a prosthetic limb. So they have implanted arrays, 4 arrays, 2 in motor area, motor cortex, and 2 in somatosensory cortex. Somatosensory cortex is the brain area which basically perceives tactile stimulation. So what they have done is, this is a 28-year-old male patient who has lost his motor skills and somatosensory cognitive skills because of a spinal cord injury. And what they are doing, they have done is that they have implanted these arrays and they have made a prosthetic limb. And they transmit the motor information from the motor cortex through these arrays to this prosthetic limb. And then they use the tactile cues from this prosthetic limb, and they feed that back to the somatosensory area, okay? And then they see that when they do this feedback thing, the motor control increases by a lot, and compared to when there was no somatosensory feedback, there was only visual feedback. Okay, does that make any sense? I don't know if I was clear enough. Can anybody say that it was clear or not? I can explain again.
Yes, it was clear. Actually, I had like kind of a reconfirming question thing that like don't we use AI to use this for people who have completely detached limbs and all, and then you know, make them work as if they were completely natural limbs?
Yes, that's what I'm talking about.
Mm-hmm. Like I have actually heard, like seen some of these things in some shows and read some
Yes,
read in some places that they say that it's like it's feeling as if it's their own limb, but
exactly.
it can't exactly like feel the touch and all.
Yes, exactly.
Mm.
So the microstimulation experiments are used for this. That they stimulate certain areas of the brain, and then they and the patients feel as if they somebody is touching their arms or like whatever, like the part of the body which they could not move or which they could not basically feel. So yeah, so this is the experiment, and I want to show you the result. So you will see two different so parallel videos. Basically, first one is when there is only the prosthetic limb is basically only controlled by motor command. So only this thing is working. And the second video when the tactile cues are fed back to the somatosensory cortex and they talk between themselves. Okay, and you'll see how different the results are. You can see the video, right?
Easy, it's perfectly clear. But
This is,
there's no audio, right?
yeah, no, no, there's no audio, sorry.
Okay.
Sorry. So these are with the feedback and these are without the feedback as written here. So you can see how quickly the task is being done when there is tactile feedback fed into the somatosensory cortex. And how precise the movements are compared to when the feedback is not present. This is supposed to be a difficult one because it requires a very precise movement. And here he's still struggling a lot. This is something which simulates pouring water from one glass to another. So these are bits of papers and plastics in these cups. The aim was to keep the glass upright. So you have to place the glass as it was before. Okay. Yeah, that was it for today. And just to clarify, for this task, in both the conditions that you saw, the vision was unobstructed. So they always had visual feedback. Anyways, the only thing that changed was the microstimulation feedback to the somatosensory cortex, which is the tactile feedback. And now you know how important tactile feedback is for your motor control. So if you can't feel things by your hand, even if you can see things and you can move your arm, but you cannot have a very precise movement. So yeah, does anybody have any questions?
Okay,
About
thank you. Yes, Dee. So thank you for the talk, first of all. And so there are some couple of questions here. So let me start with all of those.
Should I stop sharing my screen?
Yes, Dee, you may. Or I don't know. His question is the
I
episodic
think it's better if I stop sharing my screen, and the people who are having questions, they can turn on their video and ask the questions themselves.
Yes, yeah. Sure, Dee. In that case, Alokendu, you can turn on your video and ask your question directly.
Yeah.
Sorry, Dada.
Yes, you are. You were audible. Now you are not.
Hello, Alokendu, you can turn on your video and audio and ask your question directly.
If you feel
Okay.
comfortable, otherwise you can
Okay,
just
Didi.
write it.
Am I visible? Okay. First of all, thank you for this talk session. A lot of
Yeah.
information I gained
Welcome.
from this session. So, Didi, my question is that in the during our sleep, we see some of dreams. It may be horror or
Yeah.
something.
Yeah.
So the episodic memory are the play a role in this dream.
No, not really, because the dreams are that is a very different part of neuroscience. That is basically distorted feelings and distorted experiences that you see in dreams, or your anticipation, your anxiety, all of that coming together. And that basically happens in rapid eye movement cycle in your dream. I mean, episodic memory could be a part in it, but it really does not have a very significant role to play. So it could be one of the things that also contributes, basically.
Okay.
Yeah.
Okay, and my second question is that why you choose monkey for your experiment?
Monkeys,
I mean,
because the rat brain is not very comparable
no,
to
no,
human
no, no.
brains.
I want to say that there are some similar, I mean, chimpanzee or other.
Yeah.
I mean, they are similar in. So
Yeah, I got your question. So chimpanzee or the greater apes, a lot of experiments do not happen in them, so it's not allowed. uh, I mean, the greater apes are, at least in Europe, it is a thing that people don't use greater apes for experiments. And one of the reasons for that is that more you go up in the evolution tree, you have more perception of, uh, I mean, the negative feelings, as in like the pain, uh, the depression, feeling tortured, and all of that.
okay,
So
okay.
it's not allowed. I mean, it's prohibited to use greater apes for the experiments.
So next, I guess Akash had a question.
Yeah.
Hello, Didi.
Hello.
Uh, Thank you for this talk. This was very good, very informative.
Thanks
My
to
question, my question is not from this. My question is that what is the difference between the neural networks that are in our brain, and that we used it in machine learning.
Okay, so in machine learning, we do not use neurons. And the one thing which is very specific to neurons, the functioning of neuronal networks, is that the neuronal networks are way more flexible than machines, right? So if you have some, thank you, Jyoti. If you have some experiences, you can use them to modify the networks. And you do not have to open up your brain to change the networks, but you have to do that in machines, right? So that is the difference between machines and humans. I mean, machines are not that adaptive. You cannot there's no scope of online correction of the networks. Yeah, I think that is the
And
difference. Yeah.
the both networks function the same way.
Um, that is a very difficult question to answer, actually, because people have not yet been able to exactly pin down what the mechanism of the neuronal pathways are. We just have some ideas and some hints that for this function, this area is essential. If you remove this area, this function is not possible. Or when this function is executed, this area of the brain is getting lit up. And so our deductions are at that crude level yet. We do not know exactly what is happening in our brain yet. So it's still a very living field of research. That is the reason for that, right? So it's very difficult to say if machines and brains work in the same way, but I would tend to say no. And one of the reasons for that is that machines are, I think, a very simplistic model compared to a brain. Brains are way more complex.
Oh, okay, Didi, thank you.
Yeah, welcome.